Let's take a closer look at the results. Now there's two ways that you can do this. You can look at the specific Spear and Surname Project results page, or you can look at the results page on FTDNA. Now I'm going to look at the Spear and Surname Project results page because the FTDNA page doesn't have all of the DNA heritage results, so it's better to look at the Spear and Surname Project results page because it is more comprehensive. And this is what it looks like. Let's first look at the left-hand side of the table, and you can see that it's divided roughly into four broad groups: genetic family 1, genetic family 2, genetic family 3, and non-Spearin males. Now, genetic family 1 has 8 members altogether, but the first member, in fact, is the same person. One's tested with DNA heritage and one tested with FTDNA. So really we only have 7 members in that first genetic family. The second and third genetic families have 1 member each, and non-Spearin males also has 1 member. The family ID allows you to link this particular family to the traditional families table, which is table 2 in the traditional genealogy section, Spearin families identified so far. Then we have the customer code, or the KIT number, which is the unique identifying number for that particular person tested. Then we have the last name of the person, followed by the most distant known ancestor, the MDKA, together with their date of birth, date of death, and the area in which they lived. Then the next column refers to the haplogroup, and you can see that everyone in genetic family 1 belongs to haplogroup I2b1, whereas the other genetic families and the non-Spearin males belong to the haplogroup R1b1a2. More about that in another video. Then we have a series of numbers, and to explain these, let's look up first of all at the top of the table. This row here, entitled FTDNA marker order, and you can see that it runs from number 1 all the way up across the table to 111. So what is this? Well, these are the 111 markers that are tested by FTDNA, and they're divided into several panels. You have markers 1 to 12, which constitutes the first panel, and that's in purple. Then you have markers 13 to 25, which represents the second FTDNA panel, and they are in pink. And then we have markers 26 to 37, they are in green, and then we have panel 38 to 67. It's actually 3 separate panels, but I'm bunching it together here, and that's in blue. Then after that we have marker number 68 all the way up to 111, and that's the last group of markers that FTDNA currently test for. There are some additional markers that FTDNA do not test for, but which have been tested by DNA heritage. Now, just a couple of rows below, you'll see the YSTR marker name. So, for example, the first one is marker 393, the second one is marker 390, and so on. Then if we look at the body of the table, we'll see that there are rows and rows of numbers. And these numbers are the value assigned to each marker. So you'll see that all the values for the first marker, marker 393, are 15, and all the values for the second marker are 23, and all the values for the third marker are 15, and so on. If we look along the rows, we find that all 7 members in genetic family 1 are in fact identical on the first 12 markers, and the first 25 markers, but it's only when we get to the third panel of markers, marker 26 to 37, that's when we see that there is some differences in the values for each of these numbers. So for example, if we look at the values for marker 607, we see that 6 of the 7 members have a value of 14 for this particular marker, but one person has a value of 15. What is this? This is a mutation. Similarly, if we look at the values for marker 576, we'll see that 6 of the 7 members in genetic family 1 have a value of 17, but the last member has a value of 18. This again represents a mutation. And the same for marker number 570. The last member has a value of 21, whereas the other 6 members of this family have a value of 20. We also see that there are genetic mutations for the markers CDYA and CDYB, and then further along you'll see that there are mutations at marker 413B and at marker 446. If we look further along the row, we do see differences in the values for some of the markers. But these may not be true mutations. These are probably more likely due to the fact that different labs have different ways of reporting values for some of these markers. And this is something that requires a little bit more research on our part.